So welcome everyone to the Science of Pandemic Tech seminar series, a monthly series that's co-hosted by WeHealth and by the Big Data Institute at the University of Oxford. And so this seminar series is part of the fact that responding to a pandemic that we're still learning about as it goes along is hard, and getting the information to everyone who needs to know it in the tech, out of the scientific community and into the tech world and into the public health world, is something that just needs to be accelerated. And so this forum, if you haven't been here before, is really all about interdisciplinary discussion. We've had exposure scientists, we've had epidemiologists, we've had tech people, we've had ethicists, we've had user experience people, and today I'm really happy to say that we have people from my home discipline of evolutionary biology, what I was doing before this pandemic started. So that is me. This is my field. Although I'd done a bunch of other stuff too, and have pivoted to work on the pandemic. Evolutionary biology is my home and core. And that's the topic today. And I remember early in this pandemic, talking to a colleague, and he said, oh, you know, the really good thing about this pandemic is at least we evolutionary biologists aren't very important. Other people can take care of it. This virus is evolving slowly. It all seems to be just neutral demography going on. We don't need to worry about it. Luckily, this isn't a pandemic where we're important. And unfortunately, he was very wrong. And as we know now, the virus has evolved a lot. It has evolved to be more transmissible. It has evolved to be more deadly. It has evolved to at least a little bit escape immune protection, at least of natural infection, less so of vaccination so far. But it is evolving, and indeed in late 2020 with the alpha strain, we really sort of had the first recorded event where evolutionary biology was a scientific emergency. So on that note, I'm bringing in two evolutionary biologists that I admire a lot to speak to you all. So Katrina has done just beautiful work setting the stage for what's happening at the basic level. What is the natural history? What is SARS doing? You know, painting a picture of huge in-host diversity and tiny bottlenecks between, and just the basics of what this virus does in the wild. And her work influenced me very early on in the design of our app, in terms of understanding how dose responses work, and that the bottlenecks were so tiny and that people weren't getting infected by many. Well, really, in the real world, people mostly just got infected by a single virion if it wasn't a extreme high dose exposure. And so that, you know, probabilities were the way to go. So her work very much influenced me, and I'm so happy to have her here speaking. Tyler is one of these people I actually have met in person. So rare this pandemic. We shared an interest in very ancient protein evolution, and he's certainly changed track to still work on protein evolution, but not just very recent protein evolution, but even protein evolution that hasn't happened yet in the wild. So I hope these talks will be complementary, with Katrina telling a lot about how it's happening in the wild, and Tyler talking about sort of amazing technical capacities to predict what's going to happen before it even happens, which is another thing that I thought evolutionary biology would just never be able to do, too hard. And Tyler's really made incredible progress achieving what seemed impossible there. Before we move on and I give it over, we will be on vacation for July, and we're going to skip one. The August date I don't quite have for you all. It's still being pinned down. It'll probably be an event to do with Bluetooth sensing. But for September, we will be talking about strategies for presence tracing rather than proximity tracing, like the exposure notification system, but that works together. And so we'll have representatives from a variety of different distinct strategies for presence tracing solutions, and we'll have a panel of compare and contrast what they're all doing. But today's event is evolutionary biology, and with that, I'll hand it over to Katrina. Oh, one more thing before I hand over. Sorry, the Chat is closed to attendees. Please use the Q&A to ask questions. The way to go about it is the Q&A.
Great, thank you very much. Thank you for that lovely introduction. That was very nice to hear. So I will talk to you about SARS-CoV evolution. I'm going to, well, here we go. I'm going to start, I hope, nice and gently with some basic slides on viral evolution, before then talking more specifically about SARS-CoV-2 within host diversity and transmission and the paper we published in Science earlier in the year. And then I'll start talking about variants of concern, which won't be my work specifically, but how that kind of fits into the whole area. And then maybe some musing about whether we can predict the future, which will hopefully lead nicely into Tyler's talk. So what are viruses? They're infectious agents that replicate inside living cells. So we can think about them as packets of information, with the virus entering the host cell, replicating in the host cell, and then exiting the host cell. And what we can do is we can look at the genome of the virus, which is made up of 4 bases. These are actually the DNA bases, so A, C, G, and T. SARS-CoV-2 is actually an RNA virus, so there should be a U in there somewhere. But what we can do is we can look at the sequence of the viruses. Now, if we think about the human genome for a second, so all those letters, those A, C, Gs, and Ts, the human genome has about 3 billion letters. Which if you wrote those all in a volume of books would fill about 109 books. Now SARS-CoV-2 is an RNA virus, and it's actually very big for an RNA virus, but it's still only got 30,000 letters. So that would fill about 10 pages. So that gives you kind of an idea of the scale of the different scales in the genomes. But then we look at like the number of mistakes that take place when the genome replicates. So in the human genome, each time we replicate, so each time we have children, there's probably about 30 mistakes in all of those 109 volumes of books. It's incredibly low, the rate that we see mutations, basically. Whereas in RNA viruses, the rule of thumb is about 1 mistake per viral replication. It may be actually a bit less for SARS-CoV-2 because it's got a proofreading mechanism, but about 1 is a good rule of thumb. So many more kind of mistakes per page in your book, if you like. So we have our viruses, they're infecting cells, but then occasionally we might see a mutation. So here we've seen a mutation from an orange virus to a yellow virus, and then that replicates, and that bursts, and then this is like an evolutionary event, if you like. And what we can do is then we can compare viruses, either different viruses within the same individual. These are actually HIV sequences, all taken from the same individual, or we could compare viruses among individuals. So if we think about viral evolution here we've got a population of viruses, we have a mutation, that mutation spreads, we have another mutation, that mutation spreads, and we see this whole diversity. And what we can do is we could represent the diversity like this, so it's just a different way of showing those clouds of viruses. And then what we can do is we can actually generate what's essentially a family tree, in which we can work out which viruses descended from other viruses. And these family trees are what kind of evolutionary biologists will call a phylogeny. So when you see something that looks like this, and people are talking about phylogenies, this is essentially the process that's happened. And the rate of evolution in this depends on the mutation rate, and it depends on the replication rate. So the other thing we have to recognize with viruses is we actually have evolution happening at multiple scales. So we have evolution happening within hosts. So the viruses are replicating within that body, but then they're also replicating at the population scale. So viruses, you know, they can be brilliant within an individual, but if they don't manage to infect somebody else, then they're going to be a dead end. So at the within host scale, for example, we can draw these evolutionary trees throughout infection. So this is one for HIV. We've got a really long duration of infection for HIV, so we see a lot of within host evolution. And then we can also look at evolution at the population scale. So this is work that was done by Oliver Ratman and colleagues. And each color here on the phylogeny on the left represents sequences taken from a particular individual. And we can see that among the different individuals here, 1, 2, 3, 4, 5 individuals here, we can start to see kind of a between host kind of evolutionary pattern emerging. And so when we think about like kind of this interaction between the within host scale and the population scale, at the within host scale is going to be a stronger evolutionary force, if you like, when you have a long duration of infection and a long duration between infection and onward transmission. So this would be the case for viruses like HIV. Whereas for acute viruses, which are kind of have more of a boom-bust type life cycle, so they get into an individual, they replicate really fast, and then they have to transmit before our immune system manages to shut them down. We imagine here that it's your ability to transmit that's going to override really anything that's happening at the within host scale. And just kind of to mention that, so viral evolution is really very much a double-edged sword. So viruses can outrun our immune system, drugs and vaccines because they evolve so fast. But at the same time, we can use viral evolution to track how these viruses are spreading. So there's the basics. So now into the work that we've done. So this may ramp up quite quickly, but I'll try and take you through it. First of all, important to acknowledge the other people who took part in this work, particularly Matthew Hall and Tanya Golubchik. And this was very much a joint effort between the three of us and really quite a massive amount of work. So in Oxford at the Wellcome Centre for Human Genetics, we're part of the COG UK consortium. And in Oxford, we're sequencing up to 1,500 samples a week. And so I'm going to talk through work here that we did from samples collected between March and June in 2020 by Oxford University Hospitals Trust and Basingstoke and North Hampshire hospitals. And so these were samples collected during the first wave in spring 2020 in the UK. And we took samples were collected from these four centres within these hospital trusts. And so you can see here that they're mostly west and northwest of London. And what we did is we considered the consensus sequence. So this is kind of, if you take the population of viruses within an individual, remember within an individual, we've got a whole diversity of viruses. The consensus is like the average virus, if you like, within an individual. And then we looked at minority variants. So these are variants that are at above 3% frequency within that individual. I just wanted to go through this graph here, which may be familiar to some of you. This is again from another group, Kistler et al. And on the y-axis, we have CT, which is proportional to viral load. And on the x-axis, we've got the days from peak infection. And so this just gives you an idea of what the viral profile is during an infection. So early infection, viral load goes up very quickly, and then it gradually comes down again. And it's when the viral load is really high, so around about that peak, that we see most transmission. So we looked at the number, so we looked at our kind of 1,300 or so samples. And in each of those, we counted the number of minor alleles that we saw above 3% frequency. So on the x-axis here, we've got the number of unique mapped reads, which is, again, it's a proxy for the viral load. And on the y-axis, we've got the number of isNIVs per sample. And what you can see is that during that peak in viral load, we tend to have few isNIVs per sample. So this is when transmission is really likely. Whereas when we've got lower viral loads, we tend to see a lot more isNIVs per sample. And these are likely samples from later during infection, because remember, most of these samples are from hospitalized individuals. But there's a really strong caveat in this. And the caveat is this, is that when you get to low viral loads, you start to get sampling effects because you're not then picking up every single virus that's there. And this can really skew the number of isNIVs that you see. So this is, so on the bottom right graph here, we downsampled from the really high viral load samples, and you can see them. If you're downsampling by maybe 100 or 1,000-fold, it will look like you're detecting more isNIVs per sample, but this is just a statistical artifact. So because of this, we decided to only focus our analysis on those isNIVs, those variants that we saw in the high viral load samples, as well, bearing in mind it's the high viral loads, it's when viral loads are high that we see transmission anyway. And essentially this is what we found. So on the x-axis here is the number of isNIV sites we saw in a sample, and then the number of samples. So what you can see here is that most samples, we find very few isNIVs, 0, 1 or 2. And remember, this is a viral genome that has 30,000 base pairs. So of those 30,000 base pairs, we're only seeing any diversity at all at maybe 1 or 2 sites, possibly 3 or 4. And then if we looked at these kind of like these tail ends where we're seeing like 15 or more variable sites, it actually turns out that these are either co-infections or contaminated samples, and we can't tell those two apart. Then we also looked at the bottleneck size. So we had 16 epidemiologically linked household pairs. One of them we excluded because they were so different to each other, had 6 SNPs, so we excluded them as a direct transmission pair. And one pair had insufficient within host diversity to estimate the bottleneck size. But for the other 14 pairs, we were able to estimate the bottleneck size. So this is the number of viruses which found a new infection. So if you have an infection, then it's the number of ancestors that infection came from. And what we find is in most cases it's 1, right? So this is saying that 1 virus is initiating infection. That doesn't mean the exposure is 1 virus. It's just that 1 virus was the lucky virus that seeded all the other viruses that you see in the population. Interestingly, among those 14 transmission pairs, we observed 5 SNPs. So this is when the consensus, the average sequence in the donor and the recipient is different. And so this is what this graph here is showing. So on the x-axis, we've got the genome position, and on the y-axis, we've got allele frequency, and the vertical line is showing the difference between the source and recipient. And those arrows are showing those 5 cases where we see a SNP, what we call a SNP change, a change in the consensus. And what we can see is in every single case, in one individual, we see there's no diversity at the site, and then at that same site in the other individual, we do see diversity. And there's really 2 processes that can cause this. Either we have within host evolution, we have a minor variant appear, that's that blue circle, and then we have transmission of that minor variant. Because we've got a bottleneck of size of 1, then the consensus has changed then in the next individual. In the other scenario, we have transmission, and then we have within host evolution, strong within host evolution, and that within host evolution again can cause a result in the consensus. And so in both of these scenarios, we have kind of fixation in one individual and diversity in the other. So this led us to a hypothesis, and our hypothesis was that in a sufficiently densely sampled host population, we should be able to observe these processes on the phylogeny. And at least for some of these individuals, some of them who are healthcare workers, we did believe we had really, really dense sampling. So what did we do? We generated a phylogeny of all our samples. So this is the phylogeny here. As you can see, there's actually not that much diversity in SARS-CoV-2. If you remember those other trees that I showed you earlier, and there were, you could see a lot kind of these clear differences. And then what we did is we identified positions along the genome. So this is position 28580 out of 30,000 on the genome. We identified positions where we saw both a minor variance, so diversity in at least 2 samples, and we also saw this consensus change, which I was talking about before. And overall, we found that these iSNPs are phylogenetically associated with these SNP changes. So here's another example here, position 20796. So again, you can see here the red lines here. That's the consensus, and the circles are where are samples where we see a minor variant. And so we can very clearly see these kind of these patterns on the tree where we go from within, where we're seeing like the within host diversity and then the change on the phylogeny. And this really is evolution in action. We're witnessing the evolution, documenting the evolution of the virus here, as Joanna says, like in the wild. In some cases, we saw minor variants appearing multiple positions in the tree, but they weren't phylogenetically associated. We thought at the time that maybe we could use these to predict future evolution, but it turns out that these sites that we observed anyway in this study didn't really predict what we saw in the which we see in variants of concern. So it's not clear to what extent, at least looking at these sorts of analyses, that we can predict the future. So I'm just going to check the time. Okay, we're fine. So as a bit of a summary, so we have kind of limited within host diversity. Then we have these narrow transmission bottlenecks that can occasionally result in a consensus change, but usually don't. And between host evolution is dominated by genetic drifts. It's just random processes. So remember, this is what Joanna was saying, that evolution biologists were saying, well, evolution is really slow in this virus, and we probably will see evolution, but it's going to be nice and slow and gradual and all is fine. And this is indeed the conclusion we made when we submitted this paper originally to Science. Literally 2 days later, all the chat was about B117, which we now know as the alpha variant. So this was this new rapidly spreading lineage first detected in Kent in the UK. And what was interesting about this variant is it had a huge number of mutations, a lot of mutations in spike, a lot of mutations that really did change the virus. They weren't just drift, they weren't just changes which didn't do very much, which is what we were seeing in our Science paper. These mutations were clearly important and clearly doing something, and clearly enabling the virus to spread. And the hypothesis, and I think still is a good hypothesis, at least for the alpha variant and probably the beta and gamma variants, not so much the delta variant, is that they may have emerged in individuals with long duration chronic infection. So this is work from Steve Kempertal, who's in Ravi Gupta's group in Cambridge. And for example, they looked at one chronically infected individual, and they saw this huge amount of evolution within this individual. So this is an individual who was infected, I think, but for about 100 days before sadly passing away. but a large amount of evolution may be associated with the treatment that they were on. And because variants of concern have unique sets of mutations, again, it's this double-edged sword that I'm talking about, is that, you know, that once they appear and they emerge, it seems to be a very rare event, they can spread very quickly, but we can also track them using sequencing. And very soon, well, actually currently in the UK now, we're starting to deploy rapid genotyping methods. So you can identify these variants of concern really within a day or two of getting the positive PCR result. So how do we conclude? At the within host scale, I would say we have some rare SARS-CoV-2 chronic infections which may enable fit viruses to emerge and then rapidly spread at the population scale. And then at the population scale, as I was saying before, this is like a stronger force for acute infections because it's short duration of infection. So once these mutations appear, and that does seem to be very, very rare events, but once that rare event has happened, then the new virus can really spread through the population like wildfire, as we've seen. So can we predict the future? I would say in hindsight, maybe we shouldn't have been surprised by the hypothesized role of chronically infected individuals and the emergence of these variants of concern. I'm going to go out on a limb and say that as SARS-CoV-2 reaches the peak of the human adaptive landscape, so in other words, as SARS-CoV-2 gets better and better at infecting humans, evolution will be less dominated by increased transmissibility. So we will stop seeing these big leaps in transmissibility that we're seeing now with the alpha variant and then the delta variant. But I still think, and I think evolutionary leaps facilitated by chronic infection will get rarer. But I think the other thing to bear in mind is that the immunological adaptive landscape, so the environment, the immunological environment that the virus is constantly witnessing, could well shift again. We're seeing mass vaccination, mass infection. And I think this could then make chronic infection kind of a player again, if you like, in all of this, and really only time will tell. And at that, I think I would thank you for listening. Thank you very much.
So Tyler, you want to just take off immediately and we'll hold the questions until the end of both talks. If you can put on your video, share your screen.
Yep, finding all the tabs. All right, do you see my screen?
Yes.
The eternal question. All right. Great. So I'm excited to really build off of what Katrina first talked about and kind of looking a little bit more forward looking how we've been doing experiments to help us interpret SARS-CoV-2 evolution. And then I'll kind of conclude with thinking about how we might be using these techniques to predict and perhaps even prevent future evolution. And so the question that's really been motivating our work for the past year was put nicely in this New York Times headline, which is that the coronavirus is mutating. What does that mean for us? And so kind of just to elaborate on that, since the fall, we've all watched as these new SARS-CoV-2 variants with constellations of mutations have emerged across the world. And we've all really waited with bated breath to learn how these variants impact, these key phenotypes like disease severity, transmission of the virus, and the effectiveness of vaccines and therapies. And so during all of this, a lot of our focus within these variants has been on this viral spike protein. And so this is the protein that protrudes from the viral surface, and it's really a hotbed for the evolution of this virus. In particular, a key part of this viral spike protein is its receptor binding domain. And this is really the focus of our work. This receptor binding domain is the part of the spike protein that sits at the kind of the apex of this protein. And it's important for two primary reasons. The first of which is that it's the part of the virus that actually attaches to ACE2 receptors on our cells. And this binding between the spike receptor binding domain and ACE2 is what actually allows the virus to get into our cells in the first place and initiate infections. Furthermore, this spike receptor binding domain is one of the most important targets of immunity. So in particular, the receptor binding domain is the target of these antibodies that bind to this domain and inhibit its interaction with ACE2 to prevent viral infection. And these antibodies include all the antibodies that our own bodies are making via vaccination or response to actual infection. But it's also the target of therapeutic monoclonal antibodies that have been developed to treat COVID-19. So this kind of explanation of spike and the receptor binding domain highlights kind of to a first grain approximation, the key attributes that I think define the evolution of this virus. And that's that this virus is evolving to either maintain or improve infectivity while evading immunity. And so to a first approximation, these traits can partially be correlated with the ability of the virus to bind ACE2 receptors to enable that cellular infection while escaping binding by these human antibodies. And so when we think about this question about what virus evolution and what new mutations mean, in a large part, we really want to understand the balanced role that they have on the ability of this virus to initiate infection and evade immunity. So in order to do this, the emergence of each of these new variants typically sets off this cascade of what I call the reactive sets of experiments in order to understand what a new variant might mean. And so, this is a long process while we all sit here, waiting and wondering what's happening, because it requires, ordering a gene that encodes for this new virus, making the virus and starting to see what kinds of traits it exhibits, for example, in a Petri dish in the lab. Over, a few more weeks, we can begin to see what the virus variant might be doing in animal models of increasing complexity. It takes a while and a lot of debate and arguments to synthesize all of the data to try to understand what a virus might be doing. Meanwhile, the virus is probably increasing in the world and doing its thing. It doesn't really care about how long it takes us to understand. It's just going to do what it's going to do. And in many other cases, either it's taking off or it's already being replaced by a new variant of the day. And so to kind of, the reactive approach is always going to be needed. We need these really specific tests to understand what a virus variant might be doing. But our work has been trying to complement this by using a prospective approach to map the impacts of mutations so that, before a mutation is even detected, we have some ideas of what it might be doing. And so to do this, we have this experimental platform that allows us to measure the effects of all individual possible mutations that can happen within this spike receptor binding domain with respect to some of these key biochemical traits, such as receptor binding or recognition by immunity. And what this means is that once a new variant is actually observed in the wild, we already have a lookup table of the mutations that comprise that variant that allow us to say, at least with some initial ideas about what types of traits these mutations might be impacting. And this can give us really key information about a viral variant, while all of these other additional tests are ongoing. The method that we use to do this is called devutational scanning, and it's a pretty simple technique in theory, although it leverages a lot of advanced experimental techniques and instruments. But the premise of it is that we take a protein of interest. So in this case, we're looking at the spike receptor binding domain, and we make a panel of all the different mutant variants of this protein. So in our library or our panel, we have spike receptor binding domains, 4,000 different ones, each of which has a different single amino acid change in that protein. We then combine all of these proteins together in a flask, and we subject them in bulk to this laboratory selection that separates mutants on the basis of some function of interest. So we can then take our pool of mutants that are partitioned into the quote unquote high function bin versus those in the low function bin, use deep sequencing to see what mutants fall in the high function or the low function partition. And then we can look back and say, okay, great, this green mutation, E484K, decreases this particular function that we subjected the selection pressure to, while N501Y perhaps increases it. And so we've used this approach many times over to map the effects of all of these mutations with respect to many different sort of selective pressures in the lab. This includes the effects of mutations on binding to ACE2 receptor, on sort of the stability of the protein, its recognition by therapeutic monoclonal antibodies, as well as its recognition by human immune sera. So I'm going to walk you through kind of one example of the scale of the data that we get and kind of some of the lessons we can learn from it. And so here I'm showing you our demutational scanning data that determines the effects of all the possible amino acid changes at each position in the receptor binding domain. And these are our measurements with respect to binding to the ACE2 receptor. And so in this heat map, each of these squares indicates the effect of a particular mutation. So this red square tells us that mutating the amino acid D, or aspartic acid, at position 355 has a negative effect on binding to ACE2. And so there's a lot of patterns in this data, from kind of high level down to coarse grain detail or fine grain details. One kind of type of pattern that this map tells us is what types of sites in this protein are actually tolerant of mutation. So we can see there's many positions like this one here, 488, where all the possible mutations we can make here light up as bright red, which means all of them really kill this function of ACE2 binding. And so this isn't a position where we expect to see diversity accruing in SARS-CoV-2 evolution, because these mutations simply aren't tolerated. But at the same time, we see a lot of positions like this one here, 493, where many mutations are white, indicating that they're sort of neutral. They don't have an effect on this phenotype, or even blue, indicating that they improve the ability of this protein to bind to ACE2. And so these are the types of sites where we do expect to see mutations that are capable of accruing over SARS-CoV-2 evolution. We can also see specific mutations, for example, Y453F squared here, which has a strong affinity enhancing effect for receptor binding. And this was remarkable because this mutation actually took off for a little while when we saw these strains kind of emerging on this hotbed of evolution within mink farms in Europe. Even more strikingly, we see, back in June of last year, we had measured this N501Y mutation as having a strong affinity enhancing effect for receptor binding. And that preceded by several months the emergence of this particular mutation in really 3 of the first variants of concern that began to emerge across the globe. And so as soon as these variants started emerging, we already had ideas that this mutation was really key to improving its receptor binding and perhaps underlying the increases in transmissibility observed in some of these variants. An important conclusion of this side of our work is that mutations like N501Y are actually not unique. So we can see, as a few examples I've just circled here, that there are actually many mutations that, just like N501Y, enhance affinity for receptors. And so these are other mutations to potentially look out for as future variants continue to arise in the population. But less strikingly, but probably just as, or perhaps even more important, is all of this white space that we see in this map. So these are sites or mutations that don't have beneficial effects for receptor binding, but at the same time, they aren't deleterious for this trait either. And so these are the types of mutations that open up this large space of what we call neutral evolution, which the virus can simply sample without any detriment and continue to explore genetic space. And so in molecular evolution, we're from tons of historical work, we're well aware of this conclusion that neutral evolution has the potential to open up new evolutionary trajectories toward adaptive variants that might not have been accessible in a parental background. And so you can think of this, the analogy I draw here is you can think of it as sort of taking a road that goes perpendicular to your final destination, but it gives you access to an expressway that takes you directly there. And so you kind of go out of your direct way to get to where you might want to go in a different way or a faster way. And so just to reiterate, where does this virus want to go? As I highlighted in the beginning, kind of the 2 phenotypes that it's weighing, or the 2 traits that it's weighing, is its ability to maintain or improve infection while evading antibodies. And so that kind of transitions to the second part of my talk, which is how we've been using the same platform to map the ways by which this protein or this virus can evade immunity. And so just to reiterate what I said in the beginning is that this domain of the spike protein is a key target of antibodies that bind the domain to inhibit its infection. And so the first kind of types of antibodies I'll talk about are these therapeutic monoclonal antibodies that are being used to treat COVID-19. And so these are antibodies that are actually isolated typically from humans who were previously infected, but they've now been developed as drugs that are used to prevent or treat infection in newly infected individuals. So since the beginning of the pandemic, there have been 5 monoclonal antibodies that have been, approved or given authorization for treatment of COVID-19. This includes 2 antibodies from Eli Lilly, which are used either as a monotherapy or a cocktail, an antibody cocktail from Regeneron, and the newest of which is this antibody from Vir and GSK. And I think it's just important to highlight that these apart from obviously vaccines as a preventative measure, in terms of treatment for COVID-19, they're really among the most effective drugs that have been rapidly developed for COVID-19, and will continue to have a key role in the future treatment of, for example, breakthrough infections, as well as in individuals who can't mount immune responses, for example, to vaccination. However, these drugs are already showing vulnerability in the face of the evolution of this virus. And so the first clue about this happened a couple months ago when the FDA revoked its EUA approval for this bamlanivimab antibody. And even more dramatically, just last week, the US actually stopped recommending and distributing this entire Eli Lilly cocktail, despite having contracted a huge amount of production of this drug. And so to kind of see why that's the case, our approach of prospectively mapping mutations immediately told us why bamlanivimab and its cocktail with etesevimab was particularly vulnerable. So this is the same type of experiment I've been telling you about before, but this is how we show it for immune escape, where there's fewer mutations at play. And so we really use the visualization to emphasize what are the mutations that enable escape from binding by this antibody. And so in this map, I'm highlighting several of the sites in the receptor binding domain that were most relevant to this particular experiment I'm going to show you here. And then each of these letters is a mutation that we measured as enabling escape from binding from this antibody. So these are the mutations that we would expect would render this antibody ineffective. And so this is one of the Eli Lilly antibodies, bamlanivimab, and this is the other antibody that it's used with in a cocktail, LY-CoV-016. And so we can see these antibodies bind different sites on the spike receptor binding domain. And so different types of mutations enable escape from binding of one antibody or the other. But in terms of why use of these antibodies quickly became tenuous and has been halted, we can kind of layer on what types of mutants were emerging in these variants of concern and how do they correspond to what we know or what we knew even prior to their emergence were escape mutations for these antibodies. And we can see that LY-CoV-555 is escaped by kind of two of the most remarkable mutations that have been seen in these variants, the E484 mutations, as well as the L452 mutations. And so we can see on these maps, again, L452, we see R, for example, popping up here, and at E484, we see pretty much any mutation enables escape from this antibody. And then what's even worse is that the kind of trios of mutations that are happening in a few of these backgrounds, so the beta, gamma, and delta variants, are also fixing mutations at K417, which escape the other component of this cocktail. And so because these variants are accruing these multiple mutations at multiple immune recognition sites, they can escape kind of even these dual antibody cocktails if they happen to target the wrong sites that are beginning to vary across the population of viruses. So kind of fortunately, on the other hand, for the future of monoclonal antibodies, there's a lot more sites on this protein that antibodies can bind. And so we've mapped now dozens of these monoclonal antibodies with respect to where they can be escaped. And so I'm kind of summarizing all the different antibodies we've mapped here in this sort of layout that gives us sort of an anatomy of this protein domain. And we're not going to go into all the detail, but the colors of these pie charts sort of indicates where on this protein surface these different antibodies are binding. And so in squares, I put all of the different approved or authorized monoclonal antibodies that are in use. And you can see that they're largely binding within these blue regions. But there's also, for example, all of these orange regions where we have less antibodies that are being used. And so kind of diversifying our arsenal of monoclonal antibodies could make it such that even if LY-CoV-555 is ineffective against a particular variant, perhaps we could use S2X259 instead. And we could use this in sort of an adaptive manner where, depending on surveillance on the ground, we could tune what kind of antibody we're using. But on the global scale, we can also use this layout of epitope space to sort of understand what is driving this variation. And so this is the same layout of antibodies, but now I'm coloring it by the frequency of variation within the escape sites for each of these antibodies. And so in yellow and green are the antibodies where there's a high frequency of mutations escaping those antibodies. And kind of the key point I just want to emphasize here is that the variation that's accruing in SARS-CoV-2 sequences globally is actually quite sort of biased, or it's quite localized to a particular part of the protein, which is really this backside here. And so it's really these antibodies in this arc across the epitope anatomy that are most susceptible to the variation that's accruing so far. Whereas a lot of these antibodies are binding to sites on this protein that aren't yet showing variation within this pandemic. And so kind of the last data point I'll go over is maybe some indication of why that's the case. And that brings us to the section about how polyclonal antibody responses to this protein look. And so in contrast to sort of these monoclonal antibody drugs, where it's just a single antibody or maybe a cocktail of two, our bodies make this polyclonal response, which is to say that there are many different antibodies targeting many different sites on this spike receptor binding domain. However, I guess to our detriment, these antibodies aren't perfectly balanced. And so even though there might be many antibody lineages in our body, we might make more of a particular group of antibodies to a particular site than others. And so the virus can still mutate this particular site and get a partial benefit by escaping this dominant class of antibodies in this otherwise polyclonal mixture. So it's not going to completely escape immunity, but it can at least lessen its inhibition by escaping the most kind of potent component of that immunity. And so kind of the other half of this work that I've been doing has been led by Ali Greeny in the lab. And she's been using this platform to map mutations that enable escape from this type of polyclonal human sera. And I'll talk just about sort of convalescent human sera here. And so I'm just showing a selection of a few representative sera escape maps that Ali's generated. And really a key conclusion of her work is that E484 emerges as really a dominant site of polyclonal immune targeting across multiple individuals. And so this potentially explains why E484 and other sites near it on that sort of backside of the receptor binding domain that I highlighted are really accruing this variation. And it's perhaps because of this stereotyped response where basically all of our bodies are targeting this particular part of the virus. And so the strongest selective benefit for this virus is to sort of erase that surface so all the antibodies recognizing it are partially escaped. So I kind of want to finish by zooming back out a little bit and thinking more about this question about what viral evolution means for us, and then kind of thinking about how we might be able to predict and manage this evolution. So I think what I've sort of tried to highlight is how these prospective experiments that we do really do help us to understand new variation as it's occurring in the course of this pandemic. But I haven't been completely satisfied with simply interpreting variation, just because, I think the question all of us want to know is, can we not only understand what's happening, but like predict it before it happens? And I think the reason this is really difficult, I think can be shown with another classic analogy from molecular evolutionary biology, which is this concept of a fitness landscape. So a fitness landscape is a useful sort of instructive or illustrative tool that tries to emphasize that there's this vast space of possible genotypes, of possible mutations and combinations of mutations that an evolving population can adopt. And each of those genotypes, each of those sequences has what's called a fitness, right? It's this amorphous integrative concept that explains how well that genotype would survive or proliferate in a population. And so if we could be God and we could know the exact structure of this fitness landscape, we could know that the virus, if given infinite time, would reach the ultimate peak on this fitness landscape, the best sequence that it could find. But this is a really difficult task. So despite even our entire year of work, all we've really been able to do is take the wild type, the current existing SARS-CoV-2 sequence, and just map all of the first steps that could be possible on this fitness landscape. So we know a lot about the local architecture of where evolution might want to go, but we don't know what else is out there further away that these combinations of mutations could, jump to or newly explore. What's beyond that is we haven't actually measured fitness, right? Fitness is notoriously difficult to understand. Instead, what we've really done is measure, individual biochemical traits, and the effects of mutations on these biochemical traits can differ or be at odds, right? So, this might be the landscape for receptor binding, but maybe immunity is slightly different. And the fitness, or what's going to actually happen with the virus, is integrating these different traits depending on what matters for the virus, right? But the difficulty is we don't know how these different traits influence the ultimate fitness of this virus. There's this big black box that integrates the different traits that are impacted by a mutation to impact this concept of fitness. Even if we could see within this black box, it would look more like a Picasso painting than anything else that we could understand. And as Katrina alluded to, one of the most important bits is that it's going to even be changing over time. So as the population of naive hosts transitions to having more immunity because of infection or vaccination, that's going to change what types of traits the viruses care about. And so I'm sort of stuck on this realm of, we have these maps, And I'm still a little bit uncertain whether we can predict particular mutations that are going to be next. I'm loath to say this is the exact constellation of mutations that would do even better than Delta. But at the same time, I think there's some optimism that we can at least understand or predict in broad strokes what might be to come. And this can come from several different things. First of all, we can learn a lot from the past and our ability to interpret what prior variation has meant. So we've seen that in these variants, there have been these recurrent mutations, so mutations happening across multiple variants that are strengthening receptor binding and dampening antibody immunity. And so it's not hard to say, probably in the future, those types of mutations that are strengthening receptor binding and dampening antibody immunity are going to continue accruing, and we know what some of those might be. We also can look to other viruses, pandemic viruses, seasonal cold coronaviruses, or the 1918 flu, and see that, following an initial pandemic spillover, this dynamic of evolving over time to continue to escape antibody immunity and continue to maintain infectivity can continue for a really long time. And so we might not expect that this is just going to fizzle out because we're going to stop this virus's evolution, but it's probably going to transition to this type of endemic virus that we just live with our population immunity and dampening of the severity of infection. I think these maps that we're generating can still help a lot in knowing what's out there. I want to emphasize again that we've seen other mutations that haven't yet been seen in the pandemic, but we've measured in these maps that have the same types of biochemical consequences as these mutations that comprise the first waves of variants. And so the mutations that have been seen thus far aren't unique. And so I really want to emphasize, I don't think SARS-CoV-2 is done evolving, right? There's N501Y, but there's also Q4983Y, and there's Y456T. And there's all these different mutations that can continue to improve these traits or alter them, and these neutral spaces that continue to explore and to evade immunity. And so I think these variants are going to continue emerging many times over in the short and the medium and the long term, which leads us to how we can prepare for the future. I think our work really shows that we need to diversify our arsenal of monoclonal antibody therapies so that we're not relying on individual antibodies that are rendered ineffective months after they're first approved. I think one really exciting idea is that if we had real-time surveillance of the types of sequences that are present in an area or even in infecting an individual, combined with our maps about what antibodies work against a variant, this could help a lot in guiding antibody administration so that we're not using a monoclonal antibody against a variant that we know it's not effective against. And then finally, I think continuing to improve vaccine design and vaccine so that we get broader protection against variants and even other potential bat coronaviruses that can spill over is an important goal that should really continue to be pursued in earnest. So with that, I'll quickly thank Jesse as well as Allie in the lab who have worked on all of this work together. It's been a long year, but I feel like we've really learned a lot of collaborators and funding, and thank you all for listening.
Okay, and we'll start with questions and we will go over the hour, but feel free to go in and upvote the questions that you most want to see. And the way this will work is Stephanie will be first, Seifert, and I promote you to being a panelist, and then you have a chance to ask your question. Hang on, did that work? Where did Stephanie go?
Hi, these are great talks. Thank you all for your time. And Dr. Lythgoe, I just wanted to say that I really enjoyed reading your paper. And I just wanted to ask kind of your interpretation on some of the data coming from the immunocompromised patients where you have persistent infection, and what the drivers, the selective forces would be in those patients relative to the general population. So if you have someone who is immunocompromised and they have this persistent infection but are receiving treatment, the treatment so far has been either convalescent patient sera or plasma, or some of these monoclonal therapies, and that's also kind of present in the general population as well. But I just wonder, with your data showing that there's perhaps more diversity later in infection, but lower titers, that after these patients are receiving these therapeutics, that they're also having lower titers and are less likely to be transmitting virus.
Yeah, no, exactly. So I think, most transmission is occurring early in infection and really too soon for us to be seeing this kind of immune selection events that Tyler's talking about. So this is why I'm saying when we're talking about kind of new kind of adaptive variants appearing, that these are really rare events. So as you're saying, people maybe towards the tail end of their infections, or people who have really extraordinarily long infections. And again, so this is then time for the virus to kind of be evolving against the immune response, maybe crossing these adaptive valleys that Tyler was talking about, and so enabling you to kind of get this virus that you don't get by the kind of just really fast boom and bust going on, but really the virus isn't really subject to much immune pressure at all. Now, I think that there's compelling evidence that like the alpha variant did emerge in a chronically infected individual, just when you look at just the sheer number of mutations they have. But I certainly don't think that's maybe kind of a general case, and I don't think that's what we're seeing, for example, in the Delta variant. And then I think it is, potentially, in the tail end of infection, these very rare transmission events are likely to be important. I hope that answers your question.
Yeah, I think so. I think it's just, I mean, we had a patient who was chronically infected and just found that the titers were so low later on in the infection that we either couldn't isolate virus later on, or that, we had very low coverage with deep sequencing.
Yeah.
So that's wondering like how.
Yeah, no, I mean, and actually, I think even this rapid evolution, you only see it in a minority of chronically infected individuals. Most of them are probably not going to transmit. These are very poorly individuals that we must avoid kind of demonizing, for want of a better word. It just comes down to these incredibly rare events. We think about how many infections there have been globally, and then think about how many variants of concern there are. I mean, it's really tiny. So it really is about these incredibly rare events.
Great. Thank you.
Next question is from Cécile Vibou.
Hi. Yeah, thanks again for the great talks. I was just wondering, and I know it's kind of difficult, but can you speculate on the limit of transmissibility? I mean, Katrina, you said, it's going to move away from increasing transmissibility and towards escaping immunity, but when are we going to reach that threshold, and what would it be? It's purely about receptor binding. I mean, do we know what's the max binding that could ever be possible, and can we translate that into R0 over?
Tyler, do you have an answer?
Yeah, I can offer some thoughts. So I think in terms of correlating affinity to R0, that's a tall task. What I echo what Katrina said, which is, you can transmissibility, there is an upper limit, but what's really going to take over is immune selection. And in particular, immune selection can often have, minor side effects on things like receptor binding, right? The best antibody escape mutation you might be able to take, maybe it won't affect receptor binding, but in some cases it might have these mild deleterious effects on receptor binding, and you therefore need to take a different mutation elsewhere to restore that receptor binding back to where it wants to be. And we haven't dug into this in the detail I would want, but that might even be at play already in some of these variants like beta and gamma that have these K417 and E484 mutations, which are these largely antigenic mutations, but they also have N501Y, which doesn't really seem to impact antigenicity or immune recognition, but it does improve receptor binding and potentially offsets some of the deleterious effects that these K417 mutations have on receptor binding. And so I think over the long term, it's more about maintaining this equilibrium of keeping receptor binding while taking mutations to escape immunity, which maybe are slightly deleterious, but you can restore it back. Is an important dynamic, I think.
Okay, the next question is from Chase Nelson.
Hi, wonderful talks. Thank you so much. I can't see my question anymore, but I think I remember it. The first one is related to what was already asked, and that is, have you begun, this is specifically for Tyler, have you begun experimenting with perhaps combinations of two mutations that maybe haven't been observed yet in combination, like in your heat map, the blue ones, to kind of get a sense of, is the ability to increase or improve viral phenotypes like ACE2 binding reaching some sort of limit? And then sort of related to that, could you or have you provided maybe a catalog of those antibody therapies that are available, which would be least likely to promote an escape mutation that also benefits the virus in another way? So like, I would assume you want to have antibodies that target some part of the virus that can't escape to make it better. The red ones in your heat map.
Yeah, those are really interesting. So for your first question about combinations, we've done a tiny bit with like specific tests, but what we have ongoing right now, which we're just getting data back on, is we've just replicated these devutational scans entirely in some of these variant backgrounds. So like once you fix N501Y, how does that change the effects of all these other 4,000 mutations? So we're regenerating those entire maps in backgrounds like N501Y, E484K, and then the full triple mutant of B1351. And I suppose we better add Delta since it takes time to get these libraries ordered and in place. And as we keep saying, there's already new variants by the time that happens. So yeah, we're really interested in how mutation effects change across backgrounds. That's a super important dynamic in molecular evolution. And then toward the second question, designing antibodies. Yeah, I think that little anatomy of the surface of antibody binding sites, that's in the context of this paper where we're really working with one of these companies, Vir, to identify antibodies that are less susceptible to escape, both like in theory as well as what's actually happening in SARS-CoV-2 evolution, and using this prospective approach at the very early stages of antibody development to design less escapable, more robust antibodies from the get-go.
Thanks a ton.
Okay, next, Mahan Ghaffari.
Hi, thanks to the speakers. Can you hear me, by the way? Sorry.
Yes.
Okay, good. Yeah, I think Katrina partly answered my question about emergence of VOCs in like chronically infected individuals, but I want to sort of pick up on that, maybe slightly change my question now into saying like, what are your thoughts about this parallel evolution? We are seeing these VOCs independently arising in different parts of the world. And does it really need to be from a chronically infected individual? Could just having a larger population size, just having a larger outbreak, could give an opportunity for more mutations to arise, and that by itself could be an important factor in the emergence of these VOCs or not? Thank you.
Yeah, no, I think undoubtedly, and I think that's almost certainly what we've seen with the Delta variant. It's just that the alpha, beta, and gamma variants have this very unique profile of mutations, where it just didn't fit the kind of gradual evolution that we expect. Whereas the Delta variant, we see it's kind of we can see this kind of long evolutionary history, and so it is much more gradual. And I do expect, I would personally expect like kind of the gradual evolution kind of model to be the most important one. But our knowledge is constantly changing. There is this point that Tyler was saying about crossing adaptive valleys, and it may be that the longer duration of infections are needed to cross these adaptive valleys. When you have a mutation which is incredibly rare, and then if you need maybe 3 mutations together that are incredibly rare, then you can have all the infections that you like, but you'll never see those 3 mutations together. And this is why, for example, we use, triple drug therapy for HIV, for example, because the idea is that the virus can't escape all three drugs simultaneously. But yeah, I mean, it's important. It's an important question, and it's an unknown question, isn't it? Yeah.
Thank you. Thanks.
Next question is Justus Benzler.
Okay, thank you. Well, I wanted to ask you, we've seen in the past that after some time when a variant developed that had some advantages against the other ones, it would after some time replace the other ones, like now the Delta variant. And do you think that we will always have a situation like that if there is one variant that is, let's say, fitter than the other ones? After some months or so, we will almost only see this variant? Or can you expect, or should we also expect a prolonged coexistence of several variants? For example, one that is fitter in escaping immunity by vaccination, so in a vaccinated population, and another one which has the best infectiousness, so to say, in a non-vaccinated population?
That's a really interesting question. I think like over short and medium terms, I think from other viruses that circulate around the globe, we do sometimes see, I think, like coexistence of some flu lineages. I mean, like if you take I think it's H1, H3N2, I think for a while there were actually two lineages, and it was hard to know which one to put into the vaccines because either one could be the one of the year. But those are not always super stable because it only takes one die out of a lineage for it to be gone. And so over a really long time, we see the phylogenies of these viruses really be ladder-like, where there's one dominant trunk of the phylogeny with offshoots all along, but there's kind of one lineage that in the end always wins out just because of the irreversibility of death, I guess. But we're not yet thinking long term. It's been a year and a few months. So in terms of short term, I think that's a super interesting question. Maybe Katrina has more thoughts.
Yeah, I mean, so going back to the short term, I mean, I think Tyler is spot on in the longer term about what we might expect to see. In the short term, when we're seeing these replacement with variants, it's quite interesting because there's enough population susceptibility that these variants, for example, in the UK, they're not directly competing with each other. And what's happened actually is, for example, we had the alpha variant come through. In the UK, we had a strong lockdown, which was enough to suppress the alpha variant, but then the beta variant came along and shooted up. So the kind of the lockdown, the restrictions we had in the UK were not sufficient to keep the Delta variant down because it's so good at transmitting, but they were sufficient to keep the alpha variant down. So we can think about actually through kind of, again, taking the UK as an example, we've managed to basically eradicate SARS-CoV-2 multiple times. We've eradicated B.1.177. We've now basically eradicated B.1.1.7 through kind of through these lockdowns. It's just unfortunately these other variants have come through. So this is a slightly different process to what Tyler was talking about, because it's actually the social restrictions that have meant that one of these variants has gone down. It's not the viruses themselves in competition, if that makes sense.
Yes, thank you.
So Sally Otto is up next.
Thank you both for these excellent talks and also your work throughout this pandemic, all three of you. Really appreciated. So the focus of attention on spike makes a lot of sense in terms of that's what our antibodies see, and that's how the virus gets into our cell. But what I think it seems like there's a big gap in our understanding of what adaptive mutations exist in other parts of the viral genome. And, of course, the NSP6 repeated deletion in the alpha beta gamma was like, that's a potential one, but it's not in Delta. So anyway, Tyler, what are your thoughts about what are those mutations that could actually improve replication of the virus within the cells, even once it gets in there? What should we be looking out for? And are things like NSP6 deletion, are they ones that we should be watching out for in Delta?
Yeah, that's a super good point. I really clued in on spike, and a lot of the world has too, for many reasons. I think it's like the most attractive candidate to look at because we understand it's like a simple function to understand. A lot more people can work with it because you don't have to work with real infectious virus. You can work with spike in many like explanted settings. and so because of that, I think there's just a lot less understanding of some of these other genes and what they're doing. And yet, as you point out, they are also recurrently evolving. Some of the same deletions have been seen in, I think, in ORF8, I think, with like SARS-CoV-1 and SARS-CoV-2. So clearly there's something going on there. I definitely do not have the answer. I can hardly keep up with what's happening with Spike. But maybe Katrina, since she does a more unbiased whole genome monitoring approach, has more insight into kind of the recurrence of these mutations, or maybe I'm throwing her under the bus. I'm not sure.
Oh no, don't put me on the spot like that. Yeah, no, I have no idea either. I think it's a very difficult question. All I know is that for other viruses, we see selection all the way along the genome. So I don't expect it to be any different for SARS-CoV-2. I mean, I think we just, at the moment, SARS-CoV-2 is making the easy gains. So we're seeing the easy gains, we're seeing these bigger leaps, and as time goes on, the gains are going to be harder and harder for the virus to make. So the leaps, well, my prediction is they'll get smaller and smaller.
So I think we're just about, oh, sorry, Sally, do you have to follow up?
Oh, just a thank you.
So I think we're getting sort of to the end of overtime here. I've got to most of the most highly upvoted questions, but I do want to ask Katrina or Tyler if there's any questions that haven't been very upvoted, but that you want to jump in, if you can see the Q&A and answer anyway.
I think I remember a question about apps and, whether we should be, when
maybe Luca or I should answer that one.
Yeah, I mean, so this being a tech seminar series. Yeah, I remember someone asking, when we record if we're infected, we should record what the variant is, and I think very much so. First, because for surveillance, for understanding how the different variants are spreading and where they're spreading, but also in terms of some variants, they may have a longer infectious period or something like that, and therefore you may want to be asking people to self-isolate for longer, for example. That's
I will add to that. Yeah, I mean, I agree with everything you said, Katrina. And there are limits to the Google Apple protocol, and we have very limited metadata that we are allowed to attach, and that we've, we only have an integer of size 8, absolutely maximum if we repurpose everything. But I completely agree with you. I think there was evidence that there was a longer pre-symptomatic infectious period for Alpha. I'd love to know what the equivalent is for Delta. If you guys know that, that would be great. But the incubation periods and infectious periods may vary. And if we know them, then the apps could really use that information. The other thing is, I hope that we move into a world of pre-exposure prophylaxis, and that especially high-risk individuals who've had an exposure get the monoclonal antibody treatments before they test positive. And obviously, if it's before they test positive, you're not going to be able to genotype them first to tell them which monoclonal antibody to give them. So that would have to come via the contact tracing, which includes the digital alternative privacy preserving solutions. Would need that metadata in order to get them the right monoclonal antibody if we're in a world where there are multiple coexisting strains and different monoclonal antibodies in the different circumstances. So I think we are limited by the Google Apple API, but I think it would be really important to start, but of course it's all contingent on being able to genotype these things fast enough to be actionable, right? So there's still you guys are doing a great job for research purposes, but when are we going to be able to genotype these things in the real world in time to make treatment decisions? And of course that comes into it as well. Luca, did you want to? You're muted.
What I wanted to add was maybe that we have not spoken much about the phenotypic consequences, so not the molecular phenotypes, but actually the epidemiological phenotypes of these variants. This is still partly unknown. We know little about the consequences of these variants in terms of changes in symptoms. For example, now there's a lot of talk about different symptoms for the Delta variant. Nobody knows if it's the case or not right now. I think this is a whole part that we have not discussed today, and it's where maybe new studies could be more relevant in the future.
And apps with their privacy preserving ability to survey could also help with data generation. You know, if you're providing an app that provides value and that remains privacy and that also, you know, helps us learn, for example, what the incubation period is, what the infectious period is, what the symptom profile is as a function of the variant. That's not technically impossible for these technologies to do. That's something we should be aspiring to, I think. Any other questions you want to rescue?
No.
Okay. Thank you so much. We're 1/4 the hour then. I think we'll wrap up. Thank you. That was, those were 2 fantastic talks and a great set of questions. And unfortunately, we're going to be talking about SARS evolution for a while. But it's good to get people up to speed. And thank you for making that accessible.
Thanks.
Thank you so much.
And join us in August for something on Bluetooth and September for something on presence tracing.
Thank you. Bye, everyone.
Thanks.